We will talk about nature inside the lab using virtual reality. So, hi, Pavan.
Hi,
So you can
can
share your
you see
screen?
my screen?
Yeah, perfect. Yeah,
Am I audible?
yeah, that's perfect. Thank you. So you can start your presentation. Go ahead.
Uh, Hi, everyone. I am Pavan Kaushik. I work with Shannon Olsson at the National Center for Biological Sciences, Bangalore, India. So, we live in the complex real world. It's filled with clutter, there is visual targets, there are landmarks, there are horizon, and it seems like a hard place to navigate in. But if you look at the world in a new light, it's actually more than just that. There is polarization cues in the sky, there is magnetic field, there is odor plume, there is humidity, there is temperature, there is wind. Heck, there is even electric field and ultraviolet cues, which many organisms use to navigate this complex world. So they make use of all of this to navigate it. And we know some of them very well. They suck the life out of us and they eat most of our agricultural produce, but all is not bad. In their absence, the entire world will collapse. They play key ecosystem roles, one of them being pollination, and they also migrate over hundreds of kilometers and as a huge swarm. And if you want to understand how insects, primarily flying insects, how do they navigate these complex environments, how do they integrate these diverse multiple cues, multiple modalities, we have to be able to implicate causality. And there's a slight problem with that. That is, in the field, uh, it's easy to do these experiments, but it's very hard to implicate causality because of the challenges which happen there. On the other hand, in the lab, even though you can do these experiments, you can't recreate the scale of nature, be it the size, be it time scales, or the complexity itself. So people have come up with very ingenious ways. So virtual reality is a concept which is nearly half a century old, where Peter Kunz in 1960 they tethered a bee to a tiny rod and surrounded it by a drum with stripes on it. And based on the motion of the bee, they moved the drum in the opposite direction to make it feel like it's flying around. And it has come a long way. We have replaced the drum with LED panels where we can precisely give what input we want. It's also optically tracked. And we have learned a lot through this process. We have understood how insects perceive vision, how do the photoreceptors behave, how do they see vision vertical versus horizontal, and all the optomotor responses which go on in the process of insect vision. But there's a slight problem though, despite all its advantages. So for example, if you see this image is a typical scenery. And if you swap these pixels around, scramble it around, it looks like this. So it doesn't match anything like the previous image. And if you plot the amplitude sensitivities of how these are, you can see that the natural stimulus is the black line. It has a particular behavior. But if you scramble it around, it has no relationship whatsoever. There's a structure with its pairwise things. And we see this behavior at the photoreceptor level. If you record from the photoreceptor and see the information transfer rate, it's very different when it's naturalistic stimulus versus a function which mimics the uh, distribution. So it's not as simple as you just make a function which matches the distribution and you'll get the same thing. There is short range and long range interaction between them, which biological systems are tuned to extract and make sense of. And another example, if you go a bit higher, deeper in, is if you record from neurons. And as I said, you have these gratings, right? These black and white gratings. So you move it around at different velocities, and the neuron responds at at a different voltage level. And it has a tuning curve. Now, if you change the contrast of it, what do I mean? If you make the black less black and a more dark gray, and you make the white a bit less white and a bit pale gray, and you play it around, the behavior changes. And you can actually see the response curve changes. And you may say, this makes sense. It's also contrast sensitive. But if you take that exact same neuron and give them different sceneries, for example, like a understory or a canopy or a open ground, all the contrast levels and the dynamic range and the light levels are hugely different. And despite all that, no matter what you do in the same axis, the response is agnostic. It's only sensitive to velocity and not really to contrast level. And this should make sense, right? Because the organism is caring about at what velocity it's turning, and not really whether this leaf is slightly darker or brighter in this context. And the systems are tuned to pick out the relevant features in complex environments. And if you go a bit higher, you can look at behavior. For example, you keep different shapes and sizes with baited traps, and Donald Procopy and many others in 1970s and saw what shapes and sizes do insects like, and what colors they like, and what smells they like. And we see that they like particular shapes, and these black dots you see are flies which are stuck to it, and that is this, the apple fly. And that's also my model system. We know exactly what shapes and sizes and smells we like. And we thought, let's make the existing VR better from where we are right now. And how we did that is by tethering the insect to a tiny rod and giving this complex scenery. The reason we give this complex scenery is, despite the power of tiny gratings, the limitation is you can't give parallax, you can't give perspective, you can't give a sense of 3D or naturalistic features. That's a limitation. So we could give it with the new monitors, and we also have these capillaries which give it 360-degree airflow and odor. So I can give exactly what the fly wants to see, what it smells, and what it feels by airflow. And I can track the wing motion. So what do I mean? So this is the video from the camera which is coming in real time. And what I do is, if the fly wants to fly straight, it would beat both its wings equally hard. But if it wants to fly left, it would beat its right wing harder than the left, like how you're swimming, and vice versa. So if the fly says I want to turn left, what I would do is I would move the images in the monitor right. And so essentially the fly is using its wings as a joystick to control the visual input over there and playing a video game in some sense. So we measure something called gain, which is a sense of what is the ideal steering wheel sensitivity you want. And there is a range at which they behave. We call it maneuverability and stability. And this is the range RVR uses. But typically in the field, we have used quite a low number. And that's a problem, right? Imagine you're responding to a turn, but if your gain is too low, you really can't turn fast enough and you'll never be able to turn. So we hope that we use after calibrating to this, we wanted to understand how do insects navigate this complex environment. So what do I mean by complex? You have ground, you have the mountains, you have trees, and we want to see how they navigate in that. So here is a video, and I hope you can see the video. This is the arena, which gives you 360-degree visual input to the fly. In the center of that arena is a tiny fly which is stuck to a needle, which you'll see. And it has capillaries around it, which gives it airflow and wind, so airflow and odor. And what you're seeing right now is the fly controlling the scenery right now based on its wings as a joystick. So this is not a video input. It's actually the fly controlling the world. It's not a simulation in that sense. It's completely in the control of the fly. So it's weaving through, ducking and weaving through the leaves, going through the fruit, and throwing out its legs about to land. And we do this enough times. So in this example, when you give no trees, no trees, they don't go to any part of the world. So they have no preference to a particular mountain or a particular patch of grass. And we give a tree, and they straight bang go to the tree there consistently again and again. And we do not do it with just a pest. We also did it with a mosquito, which is a vector, and male mosquitoes, they also happily go to a tree. We also did with the pollinator, which goes to a flower happily. So what we have shown is multiple different species go to the relevant ecologically relevant objects in this virtual environments by looking at a bunch of pixels. And it may seem like it's not really a challenging task, but however, our best human-made creations still do a horrible job at navigating these real environments and often keep crashing because they can't do it autonomously. Now, that set aside, now let's see, how do they perceive distant targets? How do they find things which are beyond a particular distance? How do they navigate that? So I put trees at different distances. And here is what you see. So this is a heat map. So the amount of green sort of indicates the patch residence time, the number of seconds the fly was in that region. So if it is more green, that means the fly was mostly in that place. If it is white, it never went there. So as I put the trees at different distances, the chances of the probability of the fly reaching the tree reduces. And beyond a particular distance, the fly never reaches. So here I can tell you, for an apple fly, for a 4-meter tree, it will never be able to reach beyond 22 meters. I can tell you with this. This is a reactive distance in some sense, and these are useful numbers. And you may say, how is this any different from using a grating and choosing the finest grating? And then you'll ask me, what is the finest grating you can see? And I can tell you that's your acuity. But that's not exactly it. Interomotidial angles anatomy give you a sense of the the biophysical limit of it. But what is the smallest font size you can read is the more reasonable thing from a behavioral perspective, and that's what we are measuring. What is the smallest before which you won't reach the object? And that's what we are measuring in this reactive distance, which is important because when our paper just came out a couple of months ago, A researcher who works in an agricultural institute contacted us and asked us these numbers for a pollinator. And these are important to understand pollinator efficiencies. What is the optimal foraging distances they do with? And these numbers are very hard to estimate in the real world. Now, all of that was fine, but how do they do it without, how do they estimate depth? Because flies don't have any stereo vision. They don't have overlapping ommatidia in the vision field as much. So what you see here is, what if you can, what is here is two trees, and they look practically identical at the pixel level. But if you observe carefully, as you go closer to it, you're going at the same speed, you'll see that the left tree is expanding faster than the right tree. And that's because this tree is closer, and therefore the rate at which it expands, the second derivative, how fast it expands, is very different. It's much higher by geometry. So what I did is I put two trees, and they go to both the trees equally well, as you can see, two large trees. But if I put one large tree which is twice as far away, another tree which is half the size close by, you can see the difference. So these two trees at this location look absolutely identical. If I place you there, you can't tell me which is the closer tree or which is the further tree or which is the bigger or the smaller one. But the moment you start translating, the fly estimates these complex motion parallax cues of what is the object, what is the foreground from the background, and what are the objects which are expanding at what velocities, and which one is expanding at a faster rate, and actually chooses that and actually goes to the closer object. And we can actually show that they use these complex computer vision algorithms which still can't do it. Insects do it happily well, and this is the first evidence of such a thing. Now all of this is vision. What do you do without vision? And insects are in such environments all the time. For example, when you're a high flying migrant, or if you're a night migrant, you have very limited vision cues, and this is how it looks. Even if the world is turning, you can't tell much. So I put a vision scenery like this and gave it airflow. What do I mean by airflow? So these are the capillaries. So based on the fly's virtual heading, or which direction it's looking in the virtual world, with and the angle with it is with respect to the wind direction, there'll be an angle, and that angle offset is where I give the airflow here. This is the fly is flying upwind, the front capillaries give airflow. If the fly is going crosswind, the side capillaries give it, and if it's going downwind, the bottom capillaries give it, and so on and so forth. So I do that. And if I make the wind go left and they turn left and they go downwind or crosswind to a degree, if I make it go down and they make a U-turn, you can see a tiny U-turn here, and they go down and do it. If I make it go right, and they do this. So here fly airflow itself can provide directional cues. Not just that, I do it at different velocities right now. At zero velocities, the fly go in all direction, which you can see in this direction. They go in all directions. As you increase the velocity, start to go more and more downwind, and beyond some velocity, they just go mostly downwind. But mind you, they're not getting dragged downwind. It's an active decision by them to go downwind. If I put a tree at the same velocity, they happily go upwind. And these violin plots indicate the same thing. As you go more and more downwind, they are more directional. And when the tree is there, they go upwind. And you may say this is reasonable, but these are important to actually model how insects disperse and migrate, because their migrations is a function of wind velocities. And this was done 25 years ago by in the Death Valley in California. And what they released is around 500,000 Drosophila in the middle of the desert, and they caught the desert overnight in just one night of flight, 15 kilometers away. This tiny humble Drosophila flies for such a large distance. And recently, Kathy Leach and her colleagues in a recent preprint did these experiments in the same place with better quantification using computer vision, and they show very similar results. So what I showed you is they going downwind, and they going more and more downwind as you increase velocity is exactly what they show. But the difference, this was a phenomenally hard task and an extremely great job of 6 years of field work to show this fact, which our VR neatly justifies as a tiny model system to assess these behaviors easily in a month. Now this is wind and vision. So what about odor? So let's put all of them together. So what you will see right now is the fly in the virtual world. You will see a tiny green bar on the top, which indicates when the fly is receiving odor, and you'll also hear a sound. And that's a trajectory. And this is the video of the fly, which is controlling the world. So what you're seeing right now is the fly flying upwind with respect to the odor. And the odor frequency increases as it goes more and more upwind, because that's how turbulence and advection make the odor disperse in these real complex environments. And in a moment, the fly will lose the odor plume. Now it's outside the odor plume. Now it will start to meander, cast and surge, and try to reacquire the plume. You can see that in a moment. You can see these behaviors right now. And these are things which insects do often in the real world. But what we can do due to VR, as I said, so if the fly is outside the plume, it doesn't receive odor. If it's inside the plume, based on how far away it's from the target, it receives at different frequencies. Now you combine that with wind, and if you just give no odor or no wind, they don't go to the target. If you give it only odor, they don't really go either. If I give it just wind, they still don't do it. But if you combine all the wind and the odor, which we can do in the virtual world, and you can see that they go upwind and reach the target. In summary, what I've shown you is insects navigate to virtual objects in complex environments and disseminate foreground from the background, which is a very challenging task. We can measure critical ecological parameters in VR, which are very important in field studies and in pesticides and pollinators. They can also compute localized motion parallax cues for decision making, which are challenging computer vision problems. And it'll be a great venture to understand how insects do this rather at ease. They also navigate using airflow alone without any visual cues, which are important for modeling migration. And they also combine airflow and odor to follow plumes, and and it'll be important for decoupling plume following algorithms. And our VR has come a long way. And this is the Drosophila, and right now you can see a humble Drosophila right now navigating to a tree and navigating through these worlds. And we hope that with more of our arena, which is an open source arena, which any of you can build at home right now, can study your model system of your choice in its natural habitat, be it a rainforest or a savanna or a desert, and understand the diverse behaviors they do. Crossing this bridge was not easy, and a lot of people were important in doing this, without which it wouldn't have happened. Thank you.
Thank you, Pavan, for this really nice presentation. That's really super cool to see this really impressive setup. Um, so just while we are waiting for some question on the chat because there is a little delay, I have maybe one or two for you. First, we were wondering how you can take into account the visual field of the insects in your system.
So there are two parts because I'm giving a 360-degree input. So I'm covering pretty much everything the insect can ever see, except a very tiny bit on the top. So it has complete 360-degree visual input. But the color itself, that part is slightly challenging because these are computer monitors. So they're tuned to the human eyes. So the RGB of the computer monitors don't exactly align with the photoreceptors of insects. But behaviorally, we see that the behaviors match from what field data is. So it's certainly a caveat. We don't have ultraviolet cues or polarization cues, for example. But despite that, insects make do with this and navigate these worlds. So we can learn a lot from it, even with many of these shortcomings.
I see. Thank you for this answer. After we also have a more technical question. We were wondering how it was possible to actually track the movement of the wings in order to just adapt what is happening on the screen. Like what kind of technology are you using to be accurate
So
enough?
to be honest, that's actually not so challenging part actually, because this is actually I'm building on the shoulders of giants. So previously Andrew Straw and a couple of others actually built a tiny software called Kinefly where you can track the edges of the wings. So it's
Okay.
actually not too challenging because you have a camera which takes videos at 100 fps. It's actually a tiny webcam. It's a $3 web camera, and you track the blur of the wings, and based on the blur you can track it. It's relatively a very easy computer vision algorithm to track the edges of the wings, and you feed it back into the computer. The challenge was providing such complex input at such high refresh rates, and that's where we needed the computer prowess and the new gaming technologies we have right now.
Okay, thank you for your answer. So you received some congratulations in the chat, and you also received one question from Agnish Prusti, who actually asked, I mean, first who say that it was really an excellent work, and ask if the rate of retinal expansion is faster from a farther object versus a slower extension from a closer object. Will it still prefer faster expansion, which is farther?
So that's a lovely question. So now you are teasing apart size from distance and playing around with it. These are the next set of experiments I plan to do, and it's very hard to tell what's going to happen. Like, are they going to pay attention to the absolute size, or are they going to pay attention to the rate of expansion, or a combination of the two? And without experiments, I can't answer that. But those are lovely next steps.
Okay, thank you for this answer. So, like, I think we are actually running out of time for other question, but I'm pretty sure you will receive many other on your Discord channel. So do not hesitate to go on it to reply. Thanks to everyone for your attention for this talk, and we'll go for the next one. Thank you again, Pavan, for your presentation.